Hello and welcome to today's lecture on history of viral disease diagnosis. Throughout human history, infectious diseases caused by viruses have plagued population, presenting challenges for diagnosis, treatment, and prevention. In this lecture, we will embark on a journey through time, tracing the evolution of diagnostic techniques from ancient civilizations to modern science. Through this lecture, we'll highlight the contributions of pioneering scientists and researchers who pushed the boundaries of medical knowledge, often in the face of great adversity. As we delve into the history of viral disease diagnosis, we'll gain a deeper appreciation for the challenges faced by our predecessors and the remarkable progress that has been made in the fight against infectious diseases. In 1793, Philadelphia, the bustling capital of the newly formed United States, was struck by one of the most devastating outbreaks in its history, yellow fever. This deadly viral disease, transmitted by mosquitoes, spread rapidly throughout the city, claiming thousands of lives and throwing the community into chaos. Amidst the chaos, one figure emerged as a beacon of hope: Dr. Benjamin Rush. Despite the limited medical knowledge of time, Dr. Rush courageously stepped forward to treat the sick and dying, risking his own life in the process. Throughout history, humanity has grappled with the enigma of disease causation, often attributing illnesses to supernatural or mystical forces. Many ancient cultures believed that illnesses were manifestations of demonic possession or malevolent spirits, necessitating rituals or exorcisms for treatment. Next, the notion of disease as a punishment from the gods was prevalent in various civilizations. Diseases were often interpreted as divine retribution for moral transgressions, leading to rituals of atonement and supplication. Another widespread belief was in miasmic emanations, which held that diseases arose from foul odors or bad air emanating from decaying matter. This theory dominated medical thought for centuries, influencing urban planning and sanitation practices. Finally, humoralism, the dominant medical theory of ancient and medieval world, posited that disease resulted from imbalances in the four bodily fluids: blood, phlegm, black bile, and yellow bile. Treatments aim to restore equilibrium through bloodletting, purging, and dietary interventions. These beliefs, while rooted in cultural and religious context, underscored the gap between social progress and scientific understanding. The stage was set for groundbreaking discoveries in germ theory and the identification of viruses, which revolutionized our understanding of disease causation and paved the way for modern medicine. In the 17th century, a revolutionary breakthrough occurred with the introduction of improved magnifying lenses by Antony van Leeuwenhoek and Robert Hooke. These advancements in microscopy marked a pivotal moment in scientific history as they enabled researchers to explore and describe the microscopic world as never before. Antony van Leeuwenhoek, a Dutch tradesman and scientist, crafted lenses of unparalleled quality, allowing him to observe microorganisms or animalcules, as he called them, with remarkable clarity. His meticulous observations laid the foundation for the field of microbiology. Robert Hooke, an English natural philosopher, similarly made significant contributions to microscopy. His work included the publication of Micrographia in 1665, which provided detailed illustrations of various microscopic structures, such as cells, and helped to popularize the use of microscopes in scientific inquiry. The advent of these powerful magnifying lenses paved the way for further advancements in microbiology, setting the stage for one of the most transformative discoveries in the history of medicine. Louis Pasteur's work on germ theory. Louis Pasteur, a French chemist and microbiologist, revolutionized our understanding of disease causation with his experiments disproving the theory of spontaneous generation. Through meticulous experiments with swan neck flowers, Pasteur demonstrated that microorganisms did not arise spontaneously but rather came from pre-existing microorganisms. Furthermore, Pasteur's work established the concept of specific causes for specific diseases, laying the foundation for the germ theory of disease. He demonstrated that microorganisms such as bacteria and yeast were responsible for fermentation, spoilage, and infectious diseases, thereby revolutionizing medicine and public health. Koch's postulates, formulated by German physician and microbiologist Robert Koch in the late 19th century, revolutionized the field of bacteriology and provided a systemic approach for identification of causative agents of infectious diseases. Let's delve into each of these postulates. First, Koch's first postulate states that the suspected organism must be regularly isolated from the diseased organs of affected individuals and be absent from healthy organs. This criterion establishes a direct association between the presence of the organism and the manifestation of disease. Second, the organism must be capable of growth in pure culture, free from contamination by other microorganisms. This criterion ensures that observed effects are solely attributable to the isolated organism. Koch's third postulate stipulates that the isolated organism must be capable of recreating the disease when introduced into a susceptible host. This step confirms the pathogenic potential of the organism. Finally, Koch's fourth postulate requires the re-isolation of the offending organism from the experimental host, thereby completing the cycle of infection and confirming the causal relationship between the organism and the disease. In summary, Koch's postulates provide a rigorous framework for identifying the causative agents of infectious diseases, establishing a direct link between microorganisms and specific illnesses. These principles have played a pivotal role in advancing our understanding of infectious disease pathology and have contributed to the development of effective diagnostic and treatment strategies. Adolf Meyer, a German chemist, played a pivotal role in the earliest studies of viruses, laying the foundation for our understanding of these elusive entities. In 1876, Meyer embarked on a study of an unusual disease that plagued tobacco plants, leaving distinct mosaic patterns on their leaves. Intrigued by this phenomenon, Meyer set out to investigate the cause of the disease. Through meticulous experimentation, Meyer found a groundbreaking discovery. He found that the sap from a diseased tobacco plant could transmit the disease to a healthy plant. This observation hinted at the presence of an infectious agent. Initially, Meyer assumed that bacteria were responsible for the disease and attempted to filter them out of the sap. However, this effort proved unsuccessful as the disease still spread despite filtration. Faced with this puzzling outcome, Meyer reached a groundbreaking conclusion. He proposed that the disease was caused by smaller bacteria or toxins, thus challenging the prevailing understanding of infectious agents at the time. Meyer's work laid the groundwork for the study of viruses, paving the way for future researchers to unravel the mysteries of these tiny entities. His pioneering works marked a significant milestone in the history of virology and sparked a new era of scientific inquiry. In the late 19th and early 20th centuries, Dmitry Ivanovsky and Martinus Beijering independently reproduced Mayer's experiments, leading to groundbreaking insights into the nature of viruses. In 1892 and 1898 respectively, Dmitry Ivanovsky, a Russian botanist, and Martinus Beijering, a Dutch botanist, conducted experiments like Mayer's, focusing on the mosaic disease affecting tobacco plants. Ivanovsky and Beijering observed that the infectious agent responsible for the mosaic disease could pass through the filters designed to trap bacteria, indicating that it was smaller than bacteria and could not be seen under a microscope. While Ivanovsky did not pursue the matter further, Beijering was intrigued by his findings and believed that he had discovered a new type of infectious agent. Beijering coined the term soluble living germ to describe this mysterious entity. Moreover, Beijering introduced the term virus, which was originally coined in 1398 from the Latin name for poison to describe these elusive entities. This marked a significant moment in the history of virology. Rabies research has played a pivotal role in the development of comparative medicine, a field focused on studying human diseases in animals. Let's explore how the study of rabies led to the establishment of animal models for medical research. Initially, dogs were the primary animal models used in rabies research due to their susceptibility to disease. However, working with dogs presented significant dangers as they often exhibited aggressive behavior during rabies infection. In 1879, French scientist Gaultier made a significant breakthrough by introducing the rabbit model for rabies research. Unlike dogs, rabbits infected with rabies exhibited paralysis rather than aggression, making them easier to handle and study. The rabbit model offered several advantages, including shorter incubation periods and more predictable disease progression. These characteristics made rabbits a valuable animal model for studying the pathogenesis and transmission of rabies. The success of the rabbit model for rabies research laid the foundation for comparative medicine, demonstrating the utility of studying human diseases in animal models to gain insights into disease mechanisms and develop effective treatments. In their quest to combat rabies, Pasteur and Roux refined the experimental model by employing intracranial inoculation techniques, which allowed for more consistent transmission of rabies virus in animal models. This groundbreaking work led to the development of first rabies vaccine, which proved effective in preventing rabies infection in animals. The success of the vaccine marked a major milestone in the history of vaccination and public health. In 1903, Italian pathologist Adele Negri made a significant discovery that would profoundly impact the diagnosis of rabies. Negri described unique inclusions found in central nervous system of animals infected with rabies, now known as Negri bodies. Although Negri bodies were initially misinterpreted as artifacts or cellular debris, their consistent association with rabies infection soon led to their recognition as a diagnostic hallmark of the disease. This discovery revolutionized the diagnosis and surveillance of rabies, enabling more accurate identification of infected animals, facilitating timely interventions. For centuries, polio existed as a sporadic disease causing occasional outbreaks with limited understanding of its etiology and transmission. However, in the late 19th century, polio outbreaks became more frequent, prompting increased scientific interest and investigation into its nature. One notable event was the 1894 Vermont epidemic, which was the first recognized outbreak of polio. as acute nervous system disease, sparking further investigation into its neurological manifestations. In 1908, Austrian scientists Karl Landsteiner and Erwin Popper made a groundbreaking discovery. They successfully transmitted polio to monkeys, providing crucial evidence for a viral cause of the disease. This seminal discovery marked a turning point in our understanding of polio, confirming that it was indeed caused by a virus rather than a bacterial or environmental factor. By inoculating viruses into embryonated eggs, Dr. Goodpasture and his collaborators were able to study the growth and behavior of viruses in a controlled environment, paving the way for a deeper understanding of viral pathogenesis and the development of vaccines. The use of embryonated eggs in virus research became a cornerstone of virology, enabling researchers to isolate, propagate, and characterize a wide range of viruses, including the influenza virus, measles, the mumps virus, the polio virus, and so on. Peyton Roos and James Murphy set out to investigate the nature of a peculiar chicken tumor, suspecting that it might be caused by an infectious agent. In their experiments, Roos and Murphy successfully transmitted chicken tumor to healthy chickens using cell-free filtrates derived from the tumor tissue. During his research, Hurst noticed that red blood cells added to influenza A infected eggs agglutinated or clumped together, providing a visible indicator of virus infection. This observation laid the foundation of development of diagnostic tests for influenza A virus, allowing for rapid and reliable detection of viral presence. In addition to egg-based methods, serological techniques to detect virus-specific antibodies became a mainstay of viral diagnosis for many years. Working at the Pasteur Institute in Brussels, Bordet and Guingamp demonstrated the experimental basis of complement fixation reaction, which played a crucial role in our understanding of immune responses. The complement fixation reaction, as elucidated by Bordet and Guingamp, involves the binding of antibodies to antigens followed by the activation of the complement system, resulting in the formation of immune complexes. Their experiments provided a deeper understanding of mechanisms underlying immune responses and laid the groundwork for the development of other immunological assays. In a significant advancement in virology, Alexis Shalakhov and his colleagues at US National Institutes of Health developed the technique of hemadsorption. Hemadsorption involves the selective attachment of erythrocytes on the monolayer surface of tissue culture cells infected with viruses. In January 1941, the US Army took a pioneering step by founding the inaugural General Diagnostic Virology and Rickettsiology Laboratory. This laboratory was established at the esteemed Walter Reed Army Medical Center in Washington, D.C., under the guidance of Colonel Harry Flock. Viruses are too small to be seen directly with early microscopes and posed a significant challenge to virologists. Poor tissue quality further complicated the visualization of viral inclusions, hindering progress in understanding viral structures and behaviors. Aberrations in early microscopes distorted images, impeding the clarity needed for accurate observation. The correction of these aberrations was a crucial step towards achieving sharper, more detailed images vital for studying viruses. Significant advancements in condensers, oculars, and objectives revolutionized microscopy. Condensers improved illumination, oculars enhanced viewing, and objectives boosted magnification, collectively enhancing our ability to observe viruses in greater detail. These advancements catapulted virology into a new era of discovery and understanding. Researchers could now visualize viruses with unprecedented clarity. In 1892, Guarnieri bodies, cytoplasmic inclusions, were identified aiding in differentiation between smallpox and chickenpox. E. E. Teiser observed intranuclear inclusions and multinucleated cells in varicella lesions, distinguishing it from smallpox. These are Zinc smears, which is a rapid diagnostic test for various skin lesions. The presence of these suggests infection by herpes virus. Von Glam, Pappenheimer proposed a viral etiology for the cytomegalovirus inclusion disease inclusions. Diagnosis relied on histological examination until the isolation of virus in cell culture in 1950s. Urine cytology based on the presence of inclusion-bearing cells became a valuable diagnostic tool. Ernest Ruska, Max Knoll, and other pioneering scientists played a pivotal role in the development of the electron microscope. Their innovative work laid the foundation for this transformative technology which would revolutionize our understanding of the microscopic world. Unlike traditional light microscopes, which use visible light to illuminate specimens, electron microscopes utilize a beam of electrons. This electron beam allows for much higher resolution imaging, enabling the visualization of structures smaller than what is possible with light microscopy. Since its inception, electron microscopy has continued to evolve with advancements in imaging techniques and sample preparation methods. These ongoing innovations ensure that electron microscopy remains an indispensable tool for virologists in their quest to unravel the complexities of viral infections. One of the earliest triumphs of electron microscopy in virology was the imaging of the tobacco mosaic virus. This notorious pathogen, which affects tobacco plants, served as a model system for early virologists. The first electron micrographs of tobacco mosaic virus reveal its distinctive rod-like structure, marking a historical moment in virus research. These pioneering studies did more than just unveil the appearance of viruses. They laid the foundation for classifying them based on morphology and symmetry. By examining the shapes and arrangements of viral particles under the electron microscope, scientists began to categorize viruses into different groups, a fundamental step in understanding their diversity and behavior. Enders and his team were the first to recognize the significance of cytopathogenic effect as a visual aid in detection and assay of virus infections in tissue culture. Cytopathogenic effect refers to the visible changes that occur in cells infected by viruses such as cell rounding, detachment, and formation of syncytia. This observation provided a crucial tool for diagnosing viral infections as it allowed researchers to visually confirm the presence of a virus in cell cultures. Prior to their discoveries, diagnosing viral infections relied heavily on serological tests, which were often time consuming and less definitive. This discovery enabled laboratories worldwide to isolate and identify viruses directly from clinical specimens, revolutionizing the field. Robert Channer played a key role in the development of diagnostic virology. He isolated two groups of viruses associated with croup and identified respiratory syncytial virus as a major cause of serious lower respiratory tract disease in infants and children. In 1950, The Center for Disease Control and Prevention took a significant step forward by establishing the first diagnostic virology course in the United States. This initiative provided a structured platform for training healthcare professionals in the identification and diagnosis of viral infections. By offering comprehensive instruction on laboratory techniques, sample processing, and result interpretation, the CDC laid the groundwork for a skilled workforce capable of effectively addressing emerging viral threats. Since its inception, the CDC has maintained its leadership role in the field of diagnostic virology. Through continued research, collaboration, and innovation, the CDC has developed and disseminated cutting-edge diagnostic assays and protocols for a wide range of viral pathogens. One significant advancement was the development of continuous cell lines, which allowed for the serial propagation of cell cultures. Unlike primary cell cultures that have limited lifespan, continuous cell lines could be maintained indefinitely in the laboratory. This breakthrough provided researchers with a consistent and reliable platform for studying viruses over extended periods, facilitating the development of diagnostic assays and vaccines. Plague assays emerged as a powerful technique for quantifying viruses in cell cultures by allowing researchers to visualize and count individual viral plagues. This method provided precise means of measuring viral infectivity and assessing the efficacy of antiviral treatments. Plague assays revolutionized viral quantitation and became a cornerstone of virology research and diagnostic testing. Isolation of rhinovirus, the primary causative agent of common cold, marked a significant milestone in viral diagnosis. Through advancements in cell culture techniques, researchers were able to isolate and characterize this elusive virus, shedding light on its epidemiology, pathogenesis, and potential therapeutic targets. This discovery not only expanded our understanding of respiratory infections but also paved the way for the development of interventions to mitigate their impact. Organ culture techniques emerged as a valuable tool for isolating viruses from respiratory infections, including the common cold. By culturing human tissues in vitro, researchers could replicate the physiological conditions of the respiratory tract and facilitate the growth and study of viruses. This approach provided a more physiologically relevant model for investigating viral pathogenesis and evaluating antiviral strategies. Kenneth McIntosh and colleagues made a groundbreaking discovery when they detected 8 agents in the tracheal organ cultures from patients with acute upper respiratory illness. 6 of these agents exhibited morphology like avian infectious bronchitis virus. Due to their distinctive appearance resembling the solar corona and shared biochemical characteristics, these viruses formed the basis of a new group known as the coronaviruses. This seminal finding laid the groundwork for understanding the diverse family of coronaviruses, which would later include the severe acute respiratory syndrome coronavirus, responsible for the SARS outbreak in 2003. Advancements in thin sectioning techniques allowed for better observation of tissue and virus structures under the electron microscope. By slicing specimens into ultra-thin sections, researchers could examine intricate details of cellular and viral architecture with remarkable precision. This technique provided invaluable insights into the morphology and organization of viruses within host cells, facilitating a deeper understanding of viral pathogenesis. The negative staining technique developed by Brenner and Horn in 1959 significantly improved the visualization of viral morphology under electron microscope. This innovative method offered a simple and rapid approach to preparing samples for high-resolution imaging. By surrounding viruses with heavy metal salts that produce contrasting backgrounds, negative staining enhanced the visibility of viral structures, allowing for their demonstration of viral subunits and capsid symmetry with unprecedented clarity. Negative staining played a pivotal role in advancing virology by contributing to the classification of viruses based on nucleic acid type and capsid architecture. By revealing the detailed morphology of viral particles, this technique enabled researchers to identify distinct structural features characteristic of different virus families. This information proved instrumental in elucidating the evolutionary relationships between viruses and guiding efforts to develop targeted antiviral therapies and vaccines. June Almeida was instrumental in adapting electron microscopy to clinical diagnostic virology. Recognizing the potential of electron microscope to directly visualize viruses in clinical specimens, she developed novel techniques to isolate and identify viral particles from patient samples. This innovative approach marked a significant advancement in the field, providing a rapid and reliable method for diagnosing viral infections. Albert Kappikian and his team utilized immune electron microscopy as a powerful tool to visualize and confirm the presence of Norwalk virus in infected individuals. This technique involves labeling viral antigens with specific antibodies, allowing their direct visualization under the electron microscope. The collaborative efforts of Bishop, Flevet, Middleton and Kappikian not only confirmed rotavirus as a major cause of acute infantile diarrhea, but also inspired further research aimed at understanding the epidemiology, pathogenesis, and immunology of rotavirus infections. Their pioneering work continues to drive advancements in the development of vaccines and therapeutic strategies to combat rotavirus-associated diarrheal disease and improve child health outcomes globally. Albert Kuhn's pioneering work led to the development of the fluorescent antibody staining technique. This innovative method utilizes antibodies labeled with a fluorescent molecule to detect viral antigens in tissue samples. By harnessing the specificity of antibodies and the sensitivity of fluorescence microscopy, a fluorescent antibody staining enables rapid and accurate diagnosis of viral infections within hours, a significant advancement over traditional diagnostic methods. Philip S. Gardner and Joyce McQuillan further refined the fluorescent antibody technique for diagnosing respiratory syncytial virus infection in children. Respiratory syncytial virus is a common respiratory illness in infants and young children, often leading to severe respiratory distress. By adapting fluorescent antibody technique to specifically detect respiratory syncytial virus antigens in respiratory specimens, Gardner and McQuillan provided clinicians with powerful tools for rapid and reliable diagnosis of RSV infection in pediatric patients. In 1960, Rosalind Yalow and Solomon Bersin joined forces to develop radioimmunoassay, a pioneering technique for measuring minute quantities of antigens in biological samples. Radioimmunoassay combines the specificity of immunological reactions with the sensitivity of the radioisotope detection, allowing for the precise quantification of antigens in complex biological fluids. The high rate of sensitivity of radioimmunoassay proved to be particularly advantageous for blood-brain screening, where the detection of infectious agents such as hepatitis B virus was paramount. Prior to the introduction of radioimmunoassay, traditional screening methods often failed to detect low levels of hepatitis B virus antigens, resulting in the transmission of hepatitis B through blood transfusions. Radioimmunoassay's ability to detect even minute quantities of hepatitis B virus antigens significantly enhance the blood safety and reduce the risk of hepatitis B virus transmission in transfusion recipients. The introduction of radioimmunoassay for blood-brain screening had a transformative impact on blood safety and public health. By enabling the accurate detection of hepatitis B virus antigens in blood donations, radioimmunoassay helped to prevent transmission of hepatitis B transfusion recipients, thereby safeguarding their health and reducing the burden of hepatitis B related morbidity and mortality. In 1971, Peter Perlman and Eva Engvall introduced a revolutionary method in diagnostics known as the enzyme-linked immunosorbent assay. This innovative technique has since become a cornerstone of laboratory diagnosis, enabling the sensitive and specific detection of a wide range of antigens and antibodies. The term Western blot, coined by W. Neil Burnett in 1981, encapsulates a technique independently invented in 1979 by two groups of researchers, Jane Renard, Jacob Reiser, and George Stark at Stanford University, and Harry Tobin, Theophil Staley, and Julian Gordon at the Friedrich Nietzsche Institute in Basel, Switzerland. This technique, now widely used, has become a cornerstone in medical diagnostics, with diverse applications across various diseases. In recent years, Western blot has been instrumental in the diagnosis of several infectious diseases, including human immunodeficiency virus (HIV), BFC, bovine spongiform encephalopathy or mad cow disease, FIV, feline immunodeficiency virus, HBV, hepatitis B virus, and many others. In the early 1980s, George Scholar and Cesar Milston pioneered the development of monoclonal antibodies, an innovation that revolutionized viral diagnosis. This technique involved fusing antibody-producing B cells with immortal myeloma cells, resulting in hybridoma cells capable of producing large quantities of pure specific antibodies. The introduction of monoclonal antibodies revolutionized viral diagnosis in several key ways. High specificity. Monoclonal antibodies offer high specificity, resulting in fewer false positives and increased accuracy in viral detection. Consistent quality. Monoclonal antibodies ensure consistent quality and reproducibility in diagnostic assays, reducing variability and improving reliability. Unlimited quantities. Monoclonal antibody production allows for the generation of unlimited quantities of antibodies from a single cell line, ensuring a stable and sustainable supply of for diagnostic testing. Easier standardization. The use of monoclonal antibodies simplifies the standardization of diagnostic assays, facilitating comparison between different laboratories and ensuring uniformity in test results. In 1953, James Watson and Francis Crick made a groundbreaking discovery that forever changed our understanding of genetics. They built the first accurate model of double helix structure of DNA. This monumental achievement, based on the earlier work of scientists such as Avery, Chargaff, and Franklin, paved the way for revolutionary advancements in molecular biology. Their model elegantly depicted how DNA molecules are composed of two intertwined strands, forming a helical structure stabilized by hydrogen bonds between two complementary base pairs. Nucleic acid hybridization relies on the principle of complementary base pairing between nucleic acid sequences. Label probes, typically DNA or RNA molecules, are designed to be complementary to specific target sequences of interest. When applied to a sample containing nucleic acids, the probes hybridize with their complementary sequences, forming stable duplexes that can be detected using various methods. Nucleic acid hybridization has found widespread applications in clinical diagnostics. By using specific probes designed to target sequences associated with diseases or pathogens, clinicians can accurately detect and identify infectious agents, genetic mutations, and other biomarkers in patient samples. This technique has revolutionized the diagnosis of various diseases, including viral infections, genetic disorders, and cancers. One of the most notable applications of nucleic acid hybridization in clinical research was the work of Harald zur Hausen. His pioneering studies demonstrated the association between human papilloma virus infection and cervical cancer by using nucleic acid hybridization techniques to detect human papillomavirus DNA sequences in cervical cancer tissues. Zurhausen provided compelling evidence linking human papillomavirus infection to the development of cervical cancer, ultimately leading to his Nobel Prize in Physiology or Medicine in 2008. K.V. Mullis's invention of PCR represented a quantum leap in DNA amplification technology. PCR allows for the rapid and exponential amplification of specific DNA sequences from minute amounts of starting material. This technique has revolutionized various fields including genetics, forensics, and infectious disease technologies. Real-time PCR, a significant advancement in PCR technology, combines amplification and detection in a single step. This technique enables the monitoring of DNA amplification as it occurs in real time, allowing for precise quantification of target DNA molecules. Real-time PCR has revolutionized quantitative gene expression analysis, viral load monitoring, and SNP genotyping, among other applications. Multiplex PCR represents another milestone in PCR technology, allowing for the simultaneous detection of multiple target sequences in a single reaction. This technique is particularly valuable for diagnosing complex infections as it enables the detection of multiple pathogens in a single sample. Multiplex PCR has significantly improved efficiency and throughput in diagnostic laboratories, facilitating rapid and accurate diagnosis of infectious diseases. In December 2019, reports emerged from Wuhan, China, of unexplained cases of pneumonia, signaling the onset of a potentially serious public health threat. The etiology of pneumonia cases remain unknown, prompting urgent investigation and response efforts from the global health community. Dr. Zhang Yongzheng's courageous decision to release the viral genome sequence of the novel coronavirus was a pivotal moment in the early stages of the outbreak. Countries and organizations leveraged this data to develop and validate diagnostic assays for detecting COVID-19 infections, enabling timely identification and containment of the virus. On March 11, 2020, the World Health Organization declared COVID-19 a pandemic, acknowledging the unprecedented global spread and impact of the virus. This declaration served as a call to action for governments, healthcare systems, and communities worldwide to intensify efforts to mitigate the spread of virus and protect public health. The COVID-19 pandemic has spurred remarkable advancements in diagnostic testing, driven by the urgent need for rapid, accurate, and scalable testing solutions. The innovations have revolutionized the landscape of COVID-19 diagnostics, enabling enhanced detection, surveillance, and control of the virus. Point of care tests. New point of care testing technologies have emerged, allowing for rapid detection of SARS-CoV-2 infection at point of care without the need for laboratory facilities. These portable, easy to use tests enable timely identification of infected individuals, facilitating prompt isolation and containment measures. Advancements in antibody testing have improved the accuracy and reliability of serological assays for detecting antibodies against the SARS-CoV-2. These tests play a crucial role in assessing past exposure to the virus, identifying potential community, and guiding vaccine development strategies. Whole genome sequencing. Widespread adoption of whole genome sequencing has provided invaluable insights into the genetic diversity, transmission dynamics, and evolution of SARS-CoV-2. Whole genome sequence data are instrumental in tracking virus spread, detecting emerging variants, and informing public health interventions. CRISPR-Cas-based nucleic acid detection methods offer simpler, faster, and more cost-effective alternatives to traditional PCR-based testing. These innovative techniques harness the precision and specificity of CRISPR technology for rapid identification of SARS-CoV-2, RNA, enabling high-throughput testing in diverse settings. Wearable sensors equipped with advanced detection technologies have been developed for monitoring SARS-CoV-2 in the air and physiological parameters in individuals. These sensors enable real-time surveillance of viral presence and provide early warning of potential outbreaks in community settings. Remote patient monitoring platforms and breath analysis devices offer non-invasive and rapid methods for identifying COVID-19 infections. These technologies enable remote assessment of patient symptoms, monitoring of disease progression, and early detection of respiratory abnormalities associated with COVID-19. Trained dogs have been employed to identify volatile organic compounds associated with SARS-CoV-2 infection. Their remarkable olfactory capabilities enable rapid and accurate detection of COVID-19 in various settings, complementing existing diagnostic approaches. Artificial intelligence and digital technologies play a crucial role in tracking, monitoring, and analyzing COVID-19 data. These tools enable rapid data processing, predictive modeling, and decision support, facilitating proactive interventions and resource allocation. This is a brief summary. It is a timeline showing how the development of different technologies enabled standardization of methods for diagnosing virus infections. Serology and PCR subsequently enabled detection of hundreds of viruses, largely individually, while the modern high-throughput sequencing technology platforms allow detection of thousands of viruses simultaneously. This is the reference book for this lecture. Thank you.